everyone has watched it, but about 5 people have not yet, and so you need to make sure that you do that. And there's 3 questions embedded in that that are also part of your participation grade. And there was some confusion a little bit about the first question, which has to do with how do you discriminate NPH, normal pressure hydrocephalus, from Parkinson's or Alzheimer's disease, because the 3 main symptoms for normal pressure, what are the 3 main symptoms for normal pressure hydrocephalus? You can tell me the 3. Yes?
Shuffling of the feet. Urinary incontinence. Oh gosh, what is this? The third one.
Rachel?
Dementia.
Dementia, yeah.
Obviously.
So those are the 3. So Parkinson's has both shuffling of the feet and can have cognitive impairment, dementia. Alzheimer's disease has dementia, not so much the shuffling of the feet part, although eventually Alzheimer's can have some movement problems. And so the main differentiating thing is that is towards the onset, when these symptoms first start appearing, is the presence of urinary incontinence. Now, you can get urinary incontinence in the later stages of Alzheimer's disease, but that's after the disease has progressed. So really, if we're having someone that's starting to present with these types of symptoms, it's the presence of the incontinence that's really the reason that you would want to order an MRI and look to see if the ventricles are enlarged and if a shunt could easily just fix it. Good? All right. Great, so we're going to get into our sensation and perception section of this course. And I teach a course next spring, Sensation and Perception. It's one of the upper level lab courses that you need to take eventually. So you can consider doing that if you like this section. So when we talk about sensations and perceptions, we're going to talk about how physical stimuli out in our environment, like light waves or salt that's on food, interact with receptors to get turned into action potentials, neural signals. Those are the sensations now that go into your brain, and how we interpret those sensations is what we call our perception, right? Our perception of that. And so some things to be aware of is that not everybody's sensations are the same for an identical stimulus. We have differences in our physiology. We talked about this when we talked about genetics, right? So some people don't have the genes to code for a bitter taste receptor. So when 2 people put that bitter substance in their mouth, exactly the same stimulus, some people detect a sensation of bitter, other people have no sensation. So that's a really dramatic difference of that. And each person's perceptions, even if you have the same sensation that comes in, some people like spicy food, other people don't like spicy food. And so your perception of that spicy food signal that comes in is going to be different. It's going to be different, interpreted differently by your brain. And that's based on experience and the environment. All right, so for each of the sensory systems, we're going to cover 4 things that you really need to know and understand. What is the physical stimulus, right? So for vision, that's going to be electromagnetic radiation, light waves, between a certain frequency, 380 and 760. For hearing, it's going to be sound waves. Sound waves in a range between 20 hertz and 20,000 kilohertz. So you need to know that. So maybe you make a table or something with each of these 4 things. You need to know, how does that physical stimulus get turned into a sensation? And that process of converting a physical stimulus by a receptor into an action potential is called transduction. Transduction. That's an important definition that you need to know. That's why it's in red. Then for each, now that we have sensations, we have to know, okay, well, is that sensation, how do I know that that sensation is a 5,000 hertz sound or an 8,000 hertz sound? How do I know what type of stimulus it is? And that's going to be done through a process called neural coding. right? So we're going to make a code of the sensation so that our brain knows what it's receiving. So it knows that that's a sensation from a sweet receptor and not a bitter receptor in the mouth. Finally, you need to know where does it go, what part of the brain is responsible for processing and creating the perception. And there's going to be a primary area of the brain, a secondary area, and association areas. So there's really going to be three sort of categories of where it goes in the brain. All right, and so that's how we take sensations in red and turn them into perceptions in blue. All right, another sort of definition that you need to know, the law of specific nerve energies. And what this says is that each sensory nerve is labeled for that sensory system, right? It only sends input about touch in. It doesn't send input about anything else. Or only sends input about sounds, about hearing coming in. So that means that if you were to artificially stimulate that sensory nerve, your brain would interpret that's artificial stimulation as you experience that system. And this is how things like cochlear implants can work. So when someone has a problem with transducing sounds because the cochlea is not working right, we can actually put a microphone and a wire in there where the microphone detects sounds and then recreates the neural code for that sound and electrically stimulates the cranial nerve 8. And people can then hear it again. All right. If you stimulate the optic nerve, then your brain's going to interpret that information coming in as vision. Right? And I just mentioned this. Like if you stimulate the auditory nerve, you're going to hear that. And that's what a cochlear implant does. All right. So in terms of coding, there are two ways that we do this coding. Right? The first is called label line. And label line is just as the name sounds like. It means that that line or that neuron is labeled to represent a particular thing, a particular stimulus. So anytime neuron A is active, it means there's sweet taste in our mouth. That would be an example of a labeled line. And the only thing that can activate neuron A is sweet taste, nothing else. Right? The advantage of this is that the brain doesn't have to do very much to know, to interpret that sensation. It just knows, hey, neuron A is active, something sweet is in my mouth. It doesn't have to do much processing of that. But potentially you would need a lot of neurons. So I mentioned for hearing, we can detect frequencies from 20 hertz all the way up to 20,000 hertz. That's a lot of frequencies. So that would be a lot of neurons trying to get information into the brain if we had dedicated neurons for every hertz. Right? The second way that the brain accomplishes neural coding is called the cross-fiber pattern. And so here, it's the pattern of the sensation that represents the stimulus. It's the pattern of that sensation that represents the stimulus. So, markers over here. It'll work. So we have all right, so say these are two neurons. I mentioned neuron A, neuron B. What does neuron A respond to? It's label line. Just sweet. So we put sweet on the tongue, we get a bunch of action potentials. We put salt on the tongue, sour on the tongue, bitter on the tongue, we get nothing in that neuron. Only responds to sweet. Okay? Neuron B is in a cross-fiber pattern. It doesn't respond to sweet, but when we put salt on the tongue, it responds in this way. When we put sour on the tongue, it responds this way. When we put bitter on the tongue, it responds this way. So salt, it responds vigorously, continuously. For sour, it responds at a slower rate, but constantly. And for bitter, there's sort of an on-off, on-off, on-off signal. So the advantage of this is that we have one neuron now that can send information about 3 different stimuli. So we don't need as many neurons. The disadvantage is that the brain actually has to look at the pattern and interpret the pattern and know what that pattern represents. So it requires more processing power in the brain. Good? Questions about label line and cross-fiber pattern? I'll give you a spoiler alert. Most sensory neurons use both. Okay? Most sensory systems use both. So here's an example of taste, where we see sweet represented by label line, and those other 3 represented by cross-fiber. All right, so we're going to jump in. Our first sensory system is going to be the vestibular system. The vestibular system is responsible for our balance, and it detects 2 physical stimuli. 2 physical stimuli. And to do that, it needs 2 different receptors. Okay? The first stimulus is your head position relative to gravity. Your head tilt relative to gravity. To know if I'm standing upright or if I'm laying on my back. The second is your body's acceleration and deceleration. So it's a measure of movement. And in particular, the type of movement is when we speed up or we slow down. For head tilt, the receptors are called otolith organs. And there are 2 of them. And those are used to detect your position of head tilt. And then we have 3 semicircular canals. All of this is found in your inner ear. This is the cochlea here that's going to do hearing. And so the vestibular system is right next door. Because what's the name of cranial nerve 8? Yeah, vestibular cochlear nerve. So it receives information. Cranial nerve 8 is taking the information for both the vestibular system and the cochlea, the auditory system, to the brain. All right, let's talk about how the otolith organs work. The otolith organs have hair cells, sort of like modified hair cells here, with these cilia sticking up. They're embedded in this sort of gelatin layer. Right? And on top of this are pieces of calcium chloride. They're like little rocks called otoliths, essentially. And so when we're standing upright, those otoliths are being stimulated in a way. Those rocks are sort of bending these hair cells to cause signals, right? That's the transduction, that are then sending signals to the brain. And as we tilt our head back, gravity pulls those otoliths in a certain direction that causes the hair cells to bend in a certain way that we interpret that sensation as, okay, now my head is tilted back and I'm not upright. And it's all about how gravity is pulling on these otoliths to change those sensations. All right. Now, semicircular canals. They operate in a similar way. There are 3 of them. They're orientated in different planes so that we can detect acceleration or deceleration in any direction. And they're full of fluid. And when and there's a structure called a cupula sort of suspended there in the fluid. And so when we move, that fluid moves, right? So if you go get a full cup of coffee, today's National Coffee Day, by the way. So if you go get a full cup of coffee and you're going to go get on the interstate and drive home, when are you worried about spilling your coffee in the car?
You stop that.
Yeah, when you accelerate or you decelerate, that's when the fluid moves in the cup. When you're traveling 70 miles an hour, which is fast, right, down the interstate, but you're at a constant speed, that fluid's not moving, right? The fluid only moves with acceleration and deceleration, and that's what happens here. So when we accelerate, the fluid moves in one direction, bends the cupula in that direction, and when we decelerate, it moves in the opposite direction and bends the cupula in the other way. And that bending of the cupula is what's transduced. It's what's transduced as acceleration and deceleration. All right, so, and there's hair cells, again, embedded in them. So how do these hair cells actually work? And this is a pretty, I think it's a pretty cool design, biological design. So you have these hair cells, and they get taller, though each one next to the other one is a little bit taller. And they're connected to one another with an amino acid chain, and there's a ionotropic receptor that's attached to that chain. And so when the hair cells bend in one direction, the chain gets slack, and the channel closes, and there's no depolarization, nothing's coming in to depolarize it, and so we get no action potentials. When it moves in the other direction, that amino acid chain gets taut, yanks open the channel, allows potassium to come in, which is going to depolarize that and cause action potentials. So that's how moving in the different directions with acceleration and deceleration can either not send action potentials or send action potentials. So what kind of code is being sent there? This is an example of sort of both of those things happening. Grab this. Hopefully these work. Forgot to bring the markers down. All right, so let's say I have three semicircular canals. We'll just say one's for moving forward, one's for moving sideways, right? The one moving forward, that's labeled line for moving forward, or we'll just say forward, right? So anytime that semicircular canal is activated, it's going to be for this kind of movement, right? But when I accelerate, so it's going to have a baseline signal. And that's an important thing. Write that down. So don't have that on my notes. Most neurons are going to have a baseline firing rate. That means when nothing is going on, it's popping off an action potential at a sort of a constant rate. The benefit of that is that when I accelerate, I can bend in that direction, force in that direction, yanks open that channel, and we get action potentials more than baseline, right? Increase from baseline. But when I decelerate, what happens to that channel? It closes. So I actually get inhibition, and we get a decrease from baseline. So baseline allows us to have inhibition. Without baseline, if this neuron was doing nothing, then like that over there, where there's no baseline, the only thing it can do is increase its firing. It can't decrease from zero, right? So that's the importance of baseline firing rate, is that it allows us to send two signals. So this is a cross fiber pattern. Right? Because now, depending on whether the brain has to interpret, inhibition means I'm decelerating, excitation means I'm accelerating. All right. So this is the same way, the same thing that happens, right, in the otolith organs. Same sort of transduction. Bend in one direction, those hair cells open, bend in the other direction, they close. When they bend, it's actually not, it's not like a neuron. So in a neuron, the EPSP was caused by what iron? Sodium. But here, the EPSP is caused by potassium. Potassium comes in. When potassium comes in, it triggers a voltage-gated calcium channel. Calcium comes in, that hair cell releases a neurotransmitter. That's the signal for exocytosis. And we get signals going to the brain. Good? All right. So, this is what I just covered. Each otolith, the utricular saccule, are orientated in a different way to detect different types of head tilt, right? But because they have baseline activity, it can send two signals. So it's both, and the same thing for semicircular canals, send information about acceleration, deceleration in different directions. That's labeled line for the direction. But whether it's acceleration or deceleration is a cross fiber pattern through excitation and inhibition because they have baseline activity. That's neural code, right? We've covered stimulus, receptor, transduction, now neural code. Neural code is sent in a pathway through cranial nerve 8. We already talked about it. Right? The vestibular cortical areas are kind of, you don't have like a piece of your cortex that's for balance. Balance is kind of a supplementary sensory information to help us maintain our posture and maintain our balance and our movement, and know about our movement, know what our nervous system is doing. So there's some areas, specific areas that get vestibular information. The superior colliculus is one. What is it involved with? Eye tracking. And so if we're going to track something or orientate our head to a visual stimulus, we have to know about our balance and where we are relative to gravity. Right? The information also goes to the cerebellum. So remember, the cerebellum is processing sensory motor information for coordinated body movement. And I already mentioned this when we were talking about pain, but motion sickness is when you get a feeling of nausea because your vision is not matching up with your vestibular system. And the brain doesn't know how to interpret that. And so it thinks something's wrong, like you've been poisoned. And so it initiates that gastric distress, that nausea. Vestibular signals travel to the thalamus, the VP part of the thalamus. So all the sensory systems are going to go through the thalamus, right? So there are going to be different nuclei in the thalamus associated with each sensory system. And then from there, it's sent up to somatosensory cortex to help us with knowing where our arms are in space and what our body's doing, how we're moving. Yes, Rachel?
So why are some people more susceptible to motion sickness than others?
Yeah, so it's just that conflict is more troubling for their brains. Just like I said, different people, even though we have the same sensations coming in, interpret those, their perceptions are different. Yeah, so some people are more susceptible to that than others. So if you're reading a book in a car and the book's not moving, but you're on a winding road, your body's saying, hey, we're moving, we're turning, we're doing all this, but the vision system's saying, no, you're not. The book's staying still. That's where that happens. And so what is the remedy for that? So look out the window and sync up the vision with the vestibular system. Quit reading the book and sync that back up. All right, switching gears. We're just doing We're hitting the high notes here, the highlights. We're going to talk about somatosensation, so touch. There are lots of different stimuli for touch. We're going to focus on mechanical stimuli, pressure, and temperature and tissue damage. And so temperature and pain share the same system. That's why the hotter a temperature gets, you go from warm to hot to painful. All right. There's lots of different receptors for these different types of touch. In fact, for mechanical touch, we have 4 major receptors. We're going to focus on the Pacinian corpuscle as the primary one. So I want you to focus on that one. You don't have to know all the different ones. So the Pacinian corpuscle has a sensory neuron, somatosensory neuron. It's going to go to the dorsal root ganglia and go into your spinal cord and go on up to your somatosensory cortex, right? And it has this onion-like structure, layers surrounding it. And so when a stimulus is applied, if I put pressure, constant pressure, and then remove it, it bends that. And what happens is, because of the way this onion-like structure works, when we first apply pressure, this neuron bends and sends action potentials, but then as the pressure stays constant, these layers kind of absorb it, and it stops bending and stops sending a signal. And then when the pressure is removed, it bends in the other direction and sends another signal. So what this allows us to do is have a dedicated signal that tells us when something starts touching us and when it stops touching us. Now we have other, and we call that a rapid adapting or fast adapting neuron, because it adapts, it stops during a constant stimuli. Remember I said there were 4 different major types? So 2 of them are onset, offset, and 2 of them fire constantly throughout that stimulus. So we have both signals going to our brain. If we take that neuron and remove those layers, the receptor structure, and now we apply the same stimulus, we get constant, we've removed its ability to detect onset and offset. So the ability to be rapid adapting or slow adapting totally relies on the type of receptor that neuron is embedded in. Without the receptor structure, we don't get the signal. All right, so these are the 4 different mechanoreceptors. Here's the Pacinian corpuscles we talked about. They're rapid adapting. That means on, off, onset, offset signal. And their position in your epidermis, whether it's shallow towards the surface or deep, determines the receptive field. We're not going to get into that. We get into that in the spring when we talk about this in greater detail. But essentially, that allows us to have different types of perceptions of what type of pressure is stimulating that skin. So from here, the takeaway point for slide 17 is that each of the 4 mechanoreceptors, some of them are rapid adapting, which is onset, offset. Some of them are constant, slow adapting. They fire the whole time pressure is applied. And they allow for 4 different types of perception, sensations to go to your brain to inform your perception. That's the point. You don't need to know, you don't need to memorize. I'm not going to ask you, is Merkel's disc, what type of adaptation and receptive field do they have in the exam? I won't ask you that. I want you to know the concept that we have 4 different signals that each provide specific information. All right, the pathway. You already know this pathway. We already talked about it. You come in the dorsal root ganglia, go up the dorsal columns of the spinal cord. You're going to go to the VP of the thalamus. So that was the same area as the vestibular system. Because where's the vestibular system ending up? Somatosensory cortex, right? So they go to the same area of the thalamus, and they go then to the somatosensory cortex, that post-central gyrus with the homunculus. we've talked about this. We talked about this last exam section. I'm just reminding you that we do have plasticity in the brain. So if you were to lose your middle finger, then your fourth and second digit would fill in that space, take over that area of the cortex. All right. We'll talk about language. You can have pretty dramatic plasticity, right? So somebody that's born deaf, their visual system will take over that language area and allow them to use their vision for sign language in order to interpret movement as language. All right, this is the same. This is showing that not just deprivation, not just when you lose a digit, but if you stimulate it with a lot of experience, you can change that plasticity. We talked about that previously. All right, so we talked about mechanoreceptors. The second one are free nerve endings. And free nerve endings are designed to detect changes in temperature. So we have warm fibers that detect increases in temperature and cool fibers that detect decreases in temperature. And they detect damage or extreme stimuli. And they detect that as pain. So we can have increases in temperature, decreases in temperature, and pain perception through the free nerve endings. When tissue gets damaged, when your skin gets cut or burned, it releases chemicals called histamines and capsaicin, and those stimulate the free nerve endings to signal the tissue damage and signal a pain signal to the brain. If you remember when we talked about neurotransmitters, we said that pain is such an important sensory signal for us that it has its own neurotransmitter called substance P. And so when substance P comes in and is released in the spinal cord, that is a signal for pain that goes up to the brain. There are two different types of free nerve endings, A delta and C fibers. And they enter the spinal cord and they don't go up through the dorsal columns. They actually cross over to the contralateral side at the level that they come in, and they travel up in what's called the spinothalamic pathway. So we have a dedicated pain pathway in our spinal cord that's separate from touch. Separate from touch. It goes up to the thalamus, and then it goes to both the somatosensory cortex to let us know where the pain's coming from. It's coming from my hand and not my arm. And it goes to the cingulate cortex, which is part of our limbic system for the emotional component of pain. Sam, you have a question?
Yeah, I was going to ask, you said that separate pathway for pain. I was going to ask if that includes the reflex arm. So like if you burn yourself or something like that. Is that where that's located?
Yeah. So when we have an intense signal that comes in, there's a synapse here, and that's going to trigger the motor output, but also send the pain signal. We have a natural pain management system through endorphins that once pain is detected by the brain, we want to be able to turn down the pain signal, reduce it so that we can deal with fixing whatever is hurt, right? And so there are two areas called the periaqueductal gray, which sometimes I'll abbreviate PAG. So you write PAG, it's the periaqueductal gray. And the Raphe nucleus send a descending pathway down the spinal cord and release endorphins through a heteroreceptor. That's why you had to learn heteroreceptors, because this is how the pain management system works, to shut down or reduce the amount of substance P being released. This is how opiate medicines have their pain reducing effects, because they act artificially on those endorphin receptors to shut down pain. Now, when our body reduces pain, it can send a signal down just to the level of the spinal cord where my hand is sending input pain, and decrease that sensitivity because I cut my hand, but it doesn't affect the pain sensitivity anywhere else in my body, because it's only shutting down the substance P where it's coming in. And even on the other side of the spinal cord, where my right hand is not injured, if I now slam my right hand in the door trying to get out to go fix my cut, then I'm going to not have dampened pain in my other hand, because it's only because the PAG and raphe can release it only at that spot. Now, if someone had given me an opiate to control the pain, that's going to act everywhere, globally. So then it would dampen pain signals from other parts of our body. So that's the difference between drugs that are going to act globally and our body's innate system that allows it to act in just one place. As I mentioned, it's a heteroreceptor. That means it's forming, the endorphins are acting directly on that axon. It's an axon-to-axon connection. And it shuts down substance P release. So it can block it right there and reduce the pain signal going to the brain. Yes?
I know this might be a dumb question, but what makes it deadly? What in this process makes it so, like, what's the word for it? No, never mind.
Okay. All right. So any questions about somatosensory pain system, pain management? All right. So these are the review points for that part. It's time for our jam a minute. We're going to go play in the snow. Now that we're all awake again. Switching gears to the vision system. Physical stimulus. Electromagnetic radiation. That's what light is. But it's not all electromagnetic radiation. There's a lot of things encompassed in electromagnetic radiation. There's a small group of wavelengths that we call the visible spectrum, and that's from 380 to 760. And that goes from blue to green to yellow to orange to red, depending on the length of the wavelength. Short wavelengths are blue, long wavelengths are red. And so the frequency of those wavelengths is going to correspond to the color, the hue, the quality of how we perceive that light. The amplitude, how intense is the wavelength, is going to coordinate with how bright or dim the light is, the intensity. So you can have the same 450 blue light, and if the wavelength is really large, the amplitude's big, then that's a bright blue light, and if it's very small, then that's a dim blue light. Same color, intensity change. All right, how does this get transduced? So, light's going to enter the eye, go through your cornea and your lens, which is going to focus it on your retina. The retina is the back part of the eye. And it's kind of a backwards orientation, right? So the light comes in and it actually goes through a couple of layers of neurons before it gets to the receptors. It's organized this way because back here is the pigment epithelium. And these receptors, the rods and cones, use a lot of energy. They use a lot of energy to transduce light. And so having them right here by the pigment epithelium, they get all their nutrients and energy from this blood supply here. If it was flipped around and they were sticking out here, and these neurons were behind them, then it would be hard to get the nutrients and energy to those receptors. which is why it's kind of organized in the backwards like fashion. All right, so as I mentioned, there are two types of visual receptors, rods and cones. Why do we have two different types of receptors? Well, think about the world that we live in, right? What types of light situations do we have?
Black and white versus color. Black and white versus color.
Okay? I'm thinking about intensity changes. So we have daytime, when you can walk outside at noon, and if the sun's shining, it's extremely bright. And nighttime, that if you're at night and the moon's not out, it can be very, very dark. That range of intensity is so huge that we need two different systems to detect light in the dark and light when it's bright. So rods are our low light condition, our nighttime vision. They are overwhelmed in bright light. They're very sensitive because they want to detect very low levels of light. So when you walk out in daytime conditions, they get overwhelmed, they get what we call bleached, all used up, and they're not, they don't function. They're useless. Cones are named like cones because of the shape, and these are our bright light receptors. They typically are concentrated in the center of our vision, and we have different types of photopigment in those rods and cones that allows them to detect different wavelengths of light. So for rods, there's only one photopigment. So this is rods, and so they can detect blue to about orange, but notice out here where it's red, the rods really can't detect red light. So when you're using your rod vision, you're not able to discriminate between colors. You can just see sort of shades of gray because there's only one receptor. It can be low stimulation or high stimulation, but we can't interpret that as a frequency. It's just on or off essentially. And it doesn't respond to red at all. So this is like if you're trying to pick out your shirt to wear in the dark in your room, because you're trying not to turn on the light wake up your roommate, you can't tell if it's red or green or blue, right? You can just see kind of the design on it, but you can't really detect the colors. When we have our cones have three different types of photopigments. Some of the cones are short wavelength, and they only detect blues. Some of them are medium wavelength, and they detect greens and yellows. And some of them are long wavelength, and they detect yellows, oranges, and reds. So because we have three different types of photopigments and cones, we can now discriminate between what type of wavelength based on which cones are being activated. And that gives us our color vision. So we only have color vision when we're using our cones. One rod photopigment, three cone. All right, so just to kind of summarize this, right? We have two different systems for nighttime, very low level light, and daytime, very bright. All right, so the vision system is pretty confusing. It's pretty complex and confusing. I'm not going to kid around with you on that. But I'm here to help try to simplify and help you understand it in the best way. So when light comes in, it's going to go through these layers. So this is our output, the ganglion cells. Their axons are going to become the optic nerve. Okay? We have a layer of processing cells here. And then we have our rods and cones, our receptor layer back here. We already talked about why it's organized that way is so we can get nutrients to replace the photopigments as they're used up. What percent of the day do you use your visual system? Or the flip side, when do you not use your visual system? When your eyes are closed, right? right? When there's nothing coming in. So what percent of the day do you sleep? Do normal people sleep? About a third, right? 6-8 hours out of 24 hours, something like that, right? So 2/3 of the day, you're using your visual system, right? So if that was the case, right, then think about how much energy it would take, right, to use this. So the way the visual system is designed is that it's actually active and actively inhibiting. So these rods and cones are actually releasing inhibitory neurotransmitter, and in the dark, they're releasing that inhibitory neurotransmitter to block signals from going to your brain because there's no light because you're in the dark, right? And so releasing all that neurotransmitter uses a lot of energy. So we only have to do that for a third of the day when our eyes are closed. When light comes in, it's going to stop the inhibition and allow signals to go to the brain, and now we see the light. So it's really kind of a backwards thing. So the receptors turn off, and it's called the removal of inhibition. We're going to remove that inhibition, which allows action potentials to go to the brain. So in the dark, a sodium channel is open, and sodium's coming in and depolarizing that receptor, and that's causing the receptor to release that inhibitory neurotransmitter. It's actually, this sodium coming in is called dark current. It's a current that happens in the dark. When light comes in, there's a 3-step process for transduction. Transduction of light takes 3 steps. The light activates that photopigment. So if this is a short wavelength cone, that must be blue light coming in. If this is a short wavelength and red light comes in, it won't activate that photopigment. That photopigment is sensitive to the wavelength. That photopigment releases a signal. That signal shuts down the sodium channel. And if sodium stops coming in, what's going to happen? It's going to be the opposite. This is depolarized, right? Because sodium's coming in, so it's highly positive, releasing neurotransmitter. We stop that sodium come in, the charge is going to go down, it's going to hyperpolarize, and stop releasing the inhibitory signal, and then a signal can go to the brain. So we've taken light, and we've sent an action potential to the brain because we removed the inhibition. I told you it was confusing. But break it down, just think about, it makes sense from an efficiency if you think about, we only have to do the hard work releasing neurotransmitter for a third of the day. And then break this down into the 3 steps to remove inhibition. Here's another picture, sort of showing what happens in the dark on one side and what happens in the light on the other side. If we want to make it even more confusing, this is the only place that we know of in our body where glutamate, which is what's being released here, acts as an inhibitory neurotransmitter. Because remember, we say glutamate's normally the main excitatory, but in the visual system, it's doing the inhibition. Oh, it is confusing. It's really confusing. All right. Now, we have 126 million receptors in our retina. 126 million, that's a lot. There's only 1 million, which is still a lot, but only 1 million neurons in your optic nerve. And you saw how big the optic nerve was in the sheep brain. It was pretty big, right? Compared to cranial nerve 3, that was tiny, it's huge, right? So we have to take 126 million and condense it down, funnel it down to 1 million. So essentially you can think about 126 receptors all going to one neuron. We we want to have a mechanism to know like where the light is around that receptive field of 126 receptors. If this was all just light hit here, or light hit here, or light hit here, we wouldn't know where across that receptive field the light was, because we only have one output. Does that make sense? So we do a process called center surround receptive fields. Remember I said there's a layer of processing neurons? This is one job of those processing neurons. And what it does is it says, hey, take the receptors from the middle and make them a different signal from the receptors in the surround. So it could be, this is an on-center, off-surround. So when light hits the center, that neuron gets excited from its baseline. Right? But when light hits these surrounding receptors, it becomes inhibitory. So now, even though we have to take 126 receptors and put them into one output, we can get two signals, either in the center or the surround, based on this, right? So the point of having these is that we don't lose all of our ability to discriminate where that light is across those 126 receptors. We're still able to tell if it's in the center or the surround. Okay, we get convergence of the signal without losing all acuity. All right. To make it even more complicated, is that the center surround aren't always excitatory in the center and inhibitory in the surround. They can be the opposite. It can be inhibitory in the center and excitatory in the surround. It really doesn't matter. The point is we can detect if it's the center or the surround because we know what type of receptive field it is, and we get two different signals. So don't get hung up on if the center or surround is excitatory or inhibitory. It just has to be the opposite, right? We have a baseline, and then we have two signals, center and surround. They can be coded either way. All right? So, when we talk about labeled line and a cross fiber pattern, this neuron is labeled for the place on your retina where it's receiving its input. So if I see my finger up here, that's coming in and shining on a particular part of my retina, and that corresponds to a place in space. So it's labeled line for where that signal's coming in. It's a cross fiber pattern to know, is it in the center or the surround? Cross fiber pattern, center surround. Labeled line for where the retina corresponds like a map to where things are in space. And then the cross fiber pattern is within that receptive field, and I am the center or surround. Good? All right. So this is a form of a cross fiber pattern. You guys are doing good. In sensation and perception, we have two exam sections on vision, and one exam section on vestibular and somatosensory. So today we're covering like 2/3 of the semester in one lecture. So y'all are doing really good. Like I said, we're just hitting the high notes. All right, color. How do we detect colors, different colors? We've already mentioned that we have 3 different photopigments, right? That is called the trichromatic, 3, chroma, color. Because we have a short, medium, and long wavelength cone, if only the short wavelengths are being activated, what color are we seeing? Blue. If only the long wavelengths are activated, what are we seeing? Red, right? There's a second layer of processing. So we have these 3 labeled line outputs for color, short, medium, long. They come in and form excitatory and inhibitory connections in the thalamus to now switch to what we call opponent process, so that 2 colors are going to oppose each other. red versus green and blue versus yellow. And this improves our discrimination ability even better than the labeled line. All right. So the theory from trichromatic, as I mentioned, so here, the size of these receptors, short, medium, long, represents how much activity is being shown. So if we have blue light, it's primarily just activating the short. If we have green light, primarily activating the medium. Red light, primarily activating the long. Now, what if we have yellow light? What is yellow going to activate? Where does yellow fall on the spectrum?
Medium and long.
What's that? Yeah.
Medium and long.
Between the medium and long wavelength. So it's going to activate both of those. And when medium and long wavelength are equally activated, our brain interprets that signal as yellow. What happens if both short and medium are activated at the same level? What color would that be? What's short? What's medium? So what would, if both of those are activated at the same level, what color would that be? Be like a teal, like a blue-green, aqua color, right? You all getting the idea about this? Okay. Now, here's something that I think is really cool. There is no purple color. The color purple does not exist. The color purple is our brain's perception of when short and long are activated equally. And that creates the color purple. But if you look at that visible spectrum, it starts at blue and it goes to red, and there's no purple in there. There's no purple wavelengths of light. Does that blow your mind? We call it extraspectral because it's not in the spectrum. What does the color purple historically represent?
Like royalty.
Royalty, power, like in the church, the highest like bishops wore purple. So for a long time, we've known there's something special about the color purple that's unique, right? Because it really, it doesn't exist in the visible light spectrum. So if we were to take three projectors, red light, green light, blue light, if all three are equal, if we have equal stimulation of all receptors, that becomes white. If we overlap green and red, we said that becomes yellow. We overlap green and blue, that becomes teal. We overlap red and blue, purple. All right. So, the opponent process, so we have label line coding coming out of label for short, medium, long wavelengths. As I said, the opponent process theory, now they form excitatory and inhibitory connections, so we end up going from 3 signals to 4. We get better discrimination. Because now we have a blue, and the blue is inhibited by yellow light. Green is inhibited by red. Yellow inhibited by blue, and red inhibited by green. And this is the opponent process. And I can prove this to you because I can take this square and say, hey, at the top of this square be yellow, and then at the bottom be blue, and mix, fade the light in between. And what happens in the middle? Where we have equal amounts of yellow and blue. It loses its color. They cancel each other out. Because I have equal excitation and inhibition, and so they cancel each other out. Now, if you, any painters in here, artists, right? If you take yellow and blue paint and mix them together, what do you get?
You get, it's not exactly like a gray, but it's getting close to a gray because blue and orange are, like, make each other darker, but...
Someone said it over here. Like yellow and blue make what? You want this one like in finger painting? Green. That's because it's not light. This is what we call additive. paints are subtractive, and it's different things. So don't get confused on that. Hopefully I didn't just confuse you. Focus on when we mix light together, it's different than if we mix paints together, right? If you take all the paint colors and mix them together, you get what? Black, right? So if you take all the wavelengths of light and mix them together, you get what? White. It's the opposite, right? So don't get confused on that. It's different. All right. Thanks for hanging in there. We're doing good on time. Neural pathways. So we're good. Is everybody good on physical stimulus, transduction, three-step process, removal of inhibition? Neural coding. We have label line and cross fiber pattern. We talked about neural coding for the output of receptive fields, center surround. And we talked about the neural coding for recognizing colors, differentiating colors. Okay. Everything in your left visual world is going to go contralateral to your right visual cortex. Everything in your right visual world is going to go to your left visual cortex. Contralateral, just like your somatosensory system. Okay? The trick is that when something is in your got ahead of myself. All right. We have two eyes, right? And the optic nerve are going to be those axons coming out, the ganglion cells, the output. Right? They're going to come out. They're going to go to the thalamus, the lateral geniculate nucleus, or LGN of the thalamus. And they're also going to go directly to the superior colliculus and the hypothalamus. Why are we sending light signals to the superior colliculus? You guys already told me what it does.
To know where the light is coming from.
For visual tracking, how can we track something in our visual system if we don't see it, right? So visual tracking going directly there. Now, why would we send light to the hypothalamus? What does the hypothalamus do? In general, what kinds of things does the hypothalamus do?
Homeostasis, right?
Homeostasis types of things. So do any of our homeostasis systems benefit from light? Which one?
Vestibular?
Vestibular? No. Good guess though. So let's start naming the things that the hypothalamus does. Does hunger and satiety, does that have anything to do with light? No. Does reproductive things, does that have anything to do with light? What else does it do?
Fight or flight?
Fight or flight, does that have anything to do with light? Stress response? What else? How do I tell you? You gotta guess.
Does that have to do with sleep?
Yes! Sleep-wake cycle. Does that have anything to do with light? Yes, it does. It helps reset our biological clock. Our biological clock is in the hypothalamus. We'll talk about that in the next exam section. But the visual system sends information about light-dark directly into the hypothalamus to help align our biological clock, our sleep-wake cycle. Okay. I think that's everything there. All right. So this is where I was trying to be on the last slide, but I just got ahead of myself. When we have something in our right visual field, it's going to actually project onto both of our eyes. It's not like one eye does one visual field and the other eye does all of the other one. Right? If I hold my finger up here and I close my eyes, I can see my finger with both eyes. Right? So we have to consolidate that information so we don't see two images of my finger. So the nasal half of each eyeball sends its axons to the other side. So the lateral side stays ipsilateral, and the nasal or medial side crosses at the optic chiasm and sends its half of the eye's signals to meet up with the signals from the other eye and get consolidated so we only see one of our finger. And this works with depth perception. We're not going to talk about how close or far away these are, but that works with depth perception. So if they don't match up, we actually see two of something. And you guys we'll do this real quick so we have a minute. So you take your left finger, hold it about 6 inches from your nose, and then stretch your right finger out as far as you can go. Well, look at your left finger. How many of your right do you see when you're looking at your left? You look at your left, how many of your right finger do you see? Two. If you look at your right finger, how many of your left do you see? Two. Because when we focus on depth, the plane that's not in focus is falling on two different points, and we actually see two of that image. All right? All right. Same thing for the left. It's going to come in, and the nasal or medial side is going to cross over. Right? So this is the way this sort of pathway looks, and it's the medial halves of each eye that are crossing at the optic chiasm, which is why I wanted you to know what the optic chiasm is. All right. That then consolidates our left world to our right occipital lobe, and our right world to our left occipital lobe. This is just another example of how that works. The area of the thalamus is called the LGN. I already mentioned that. There it is again. And we have, this is the primary pathway, and this is our primary cortex, occipital lobe, the primary V1, the primary visuals cortex. We have two secondary pathways that do very specific things. Superior colliculus for eye tracking or head orientation, and the hypothalamus for sleep-wake cycle. All right, so V1 is organized. Remember, how much of the brain is for visual processing? Okay. We have all of this area as vision, and then remember we said there's action going up here, and we have identification going down here. So there's a lot of our brain, maybe half of the cortex is involved with some form of visual perception. Right? Very complex. Primary visual cortex is organized in columns, and each column responds to a different angle of lines at a particular place in space. And they're organized left eye, right eye. That's where they're matching up so that I have those two signals from my left eye and my right eye that represent the one finger in space right next to each other. So both of those will fire at the same time when that stimulus is present. So our receptive fields from the eye, coming out of the eye, are those donut center surround receptive fields. When they get here, through a process of divergence, the receptive fields change from those donut center surrounds into stimuli, in particular angles of lines. This is going to detect features. So we got to take those action potentials that are really complex in our eye and reassemble them and associate color with them and sort of put back together what we're seeing. So these features that are detected in the columns are things like line orientation, angles, and simple movements. Secondary cortical areas. This is the first time we've kind of talked about secondary cortical areas because we didn't talk about them with the stimulus sensitization. We move out of V1 into these other areas that are called extrastriate cortex, and there's particular areas that do things. So V2 and V3 take the signals of those different angles of lines and reassemble them into a rectangle. And then that rectangle then goes on to, well, it has color associated with it, which is V4. If it's not moving, it's not activating that system. And as it comes out of here, it's going to go into the temporal lobe, and you're going to say, hey, I recognize that shape. That shape is an eraser. I've seen that before. If you've never seen an eraser before, you won't know what it is. You'll have to ask somebody and say, oh, that's an eraser. And then that gets coded in your temporal lobe as a memory. And anytime you see that shape, that object, it's going to stimulate that memory and identify what that object is. V5 and the middle superior temporal gyrus are movement, detecting movement, associating movement with objects. And we have two streams of processing, as I mentioned. Coming out through the parietal lobe is, how do I respond to the stimulus? Do I need to make a decision about what I'm seeing? Going down into the temporal lobe where your memories are is, what is this stimulus? Identification. So the dorsal stream is an action because you're headed towards the frontal lobe where you're going to make decisions and have movement, right? In the motor system. The ventral stream into the temporal lobe is, what is that object? Does it stimulate a memory so I know what that object is? There's a special part of the ventral lobe called the fusiform gyrus, and that's where we detect faces. Faces are such an important stimuli to us that we have a whole area of the brain just dedicated to detecting and remembering and associating things with people's faces. All right. When this area gets damaged, it affects our ability to detect faces. So I'm going to post this video. We're out of time. I'm going to post this video to the middle page. Please watch it. It's only a couple minutes long, but it's very interesting. It's about Terry Sweeney, who has face blindness because her fusiform gyrus got damaged. And this is a condition called proboscidosia, and she cannot recognize faces. All right.